Hello students. In the last lecture we have got idea about what is germplasm, which are the methods we have to use for the conservation of a germplasm. Now in this lecture today this is second lecture for the unit 3 or third unit from your plant physiology, that is germplasm conservation and cryopreservation. My friends, you have to keep in mind actually what is the necessity of conservation of a germplasm. That is also most important, isn't it? What is the necessity of a conservation of a germplasm? My friends, in the last first lecture of this unit we have got idea about the what is germplasm. Once again I am going to repeat, germplasm is a hereditary material transmitted to the offsprings through the germ cells. That is actually so. The last lecture we have got idea that is a total content of a gene which is transmitted from one generation to another generation, and whenever such a things are going to take place, that is with the help of through a germ cell, then germ cells are nothing but the germplasm, isn't it? And the very objective of germplasm conservation is to preserve the genetic diversity of a particular plant or genetic stock for its use at any time in future. Friends, also we have got idea about in situ conservation, ex situ conservation, in vitro conservation. So this slide gives us idea about what is the difference between in situ conservation and ex situ conservation. Actually, in case of in situ conservation and protection of a biodiversity takes place in its natural habitat. In case of ex situ conservation, outside the natural habitat, means artificial habitat also we can say. So in case of in situ, natural habitat; in case of ex situ, that is outside their natural habitat. Now in case of in situ, helps in a recovering population, helping them to develop their distinctive features. In case of ex situ conservation, helps in recovering population in order to prevent their extinction, isn't it? Now in case of in situ conservation, some examples which places we have to say that are the in situ conservation places, that are the national parks, biospheres, reserves, wildlife sanctuaries. These are the examples of a in situ conservation. Now ex situ, that is botanical garden, zoological parks, then safaris are the places, or we can say that our garden around the bungalow also, that is an example of ex situ conservation. What are the advantages of in situ conservation or in conservation in a natural condition? Not only conserve the genetic diversity, it also allows evolution to the genetic with climate. But in case of ex situ conservation, it does not, or it not allows the evolution to the genetic with the climate, because in natural condition definitely there is changes in the climate takes place, and according to get adjust with that one, they are changing their genetic evolution we can observe into that one. But in a artificial condition, the conditions are very much fixed, so evolution never takes place into that one. Another advantage of a in situ conservation, that it allows new alleles and new gene combination would appear with the time. As time changes, as times passes, there is a, we have to say that new combinations, new alleles and new gene combinations can be appeared. But no any type of a new gene combination occurs due to the fixed environment. Already I have told you, actually the in situ conservation is a natural conservation. Ex situ conservation, that is a artificial conservation outside the natural one. Now what is the demerit of a in situ conservation? That it's difficult to establish and very difficult to maintain, very prone or biotic, so costly that one. Again, it required costly and well occupied laboratories, where facility having laboratory. So this is the little bit difference also you have to keep in mind in case of in situ and ex situ conservation. Now my friends, as in the last lecture, means first lecture of this topic or unit, we reached up to the 4 approaches of in vitro conservation in germplasm. That is, first one, we have to say that is a cryopreservation, then cold storage, low pressure storage means LPS, low pressure storage, and LOS, that is a low oxygen storage is there. Now, cryopreservation, very important methodology this one. Cryo means you have to keep in mind, cryo means you have to keep in mind very cold. Cryo, preservation at a very cold temperature, so minus 196 degrees centigrade. We have to preserve the germplasm at a minus 196 degrees centigrade conditions are there. So that we have heard about the cryogenic engine in case of the satellite launching. They always apply the cryogenic engine. Cryogenic engine helps to maintain the temperature of a satellite when it gets launched. When it gets launched. So you have to keep in mind. So cryopreservation means preservation in a frozen state. The principle involved in cryopreservation is to bring the plant cell and tissue culture to a zero metabolism or no dividing state by reducing temperature in the presence of a cryoprotectant. Keep in mind, we are going to use some chemicals in cryopreservation, and the chemicals, whatever or substances used to protect the germplasm at very low temperature, that chemicals are known as a cryoprotectants. Am I right? But what the role is played by these, we have to say that a frozen atmosphere, a minus 196 degree temperature, that when we put a plant material or germplasm at that low temperature or frozen temperature, the cell becomes a growthless, means no metabolism takes place, no dividing of a cells takes place. Although the meristematic cells are there, they remain as it is only in a live condition. Keep in mind, my friends, once again I am going to give you idea about the cryopreservation. Cryopreservation means preservation in a frozen state. The principle involved in the cryopreservation is to bring the plant cell and tissue culture to the zero metabolism, ZERO, zero metabolism, you have to keep in mind, or non-dividing states. That is done with the help of keeping germplasm at low temperature. So the various temperatures are there. Broadly means over a solid carbon dioxide also we can use minus 79 degrees centigrade is developed due to this solid carbon dioxide, means cryoprotectant. Low temperature deep freezer at minus 80 degrees centigrade. In vapor phase nitrogen at minus 196 degrees centigrade, and in liquid nitrogen minus 196 degree temperature is there. My friends, which mechanisms is there in case of this one? I will give you idea. The technique of freeze preservation is based on the transfer of a water present in the cell from liquid to the solid state. In cryopreservation, when we put actually germplasm or living cells at minus 196 degrees centigrade or zero metabolism, we have to say that at that time the liquid which is present into the cell, that is converted into a solid state, and due to the presence of salts and organic molecules in the cells, the cell water requires much more lower temperature. We know that why minus 196 degrees centigrade temperature is required for putting that cell to the zero metabolism, because we know that in case of plant cell, the water present into the plant cell also shows presence of a salts. Organic molecules are there, proteins are there, carbohydrates are there, many more such things are there, lipids are there, and because of that one that required a more and more low degree temperature for its conversion of a liquid present into the cell into the solid one. So you'll have to keep in mind when metabolic changes takes place at that time, these things we will be able to observe. Now, precautions for the successful cryopreservation, which type of we have to take, that are the formation of ice crystal inside the cell is takes place should not be prevented as cause the injuries to the cell organelles and cell. That is most important precaution we have to take. Another, that is a high intercellular concentration of a solutes may also damage the cells. High intercellular concentration of a solutes means I have to say that proteins are there, amino acids are there. These solutes may can damage, that is to be avoided. Sometimes certain solutes from the cell may leak out during the freezing. So this also precaution we have to take, that should not happen for the successful cryopreservation. Cryoprotectants also affects the chemicals whatever we are using, that may can affect to the viability of the cells. The physiological status of the plant material is also important. Now, which type of a technique is used in case of cryopreservation? Means steps involved into the cryopreservation after getting idea about mechanism. Development of a sterile tissue, first one. Second step, that is addition of a protectant and treatment. Third one is a freezing storage. Fourth one, thawing. Last one, reculturing. Then measurement of a survival viability in plant. Regeneration is there. Now, my friends, this sketch actually diagram gives you idea about or outline of the protocol of the cryopreservation of a shoot, how the things are going to happen. As we know that here we are interested in case of germplasm conservation, we are interested in case of a germplasm preservation. So protocol or sketches, this diagrammatic representation will give us idea how the things are going to happen here. So you can see my friends in the sketch actually some shoot tips in the semi-solid material are there, that we have to shoot tip culture is there, then shoot tips we have taken, that shoot tips are pre-grown on a medium with a DMSO, dimethyl sulfosalicic acid is there. Now that when cryoprotectant, this DMSO is a cryoprotectant, in that one we have put again shoot tips plunge into the liquid nitrogen and shoot tip medium at 25 degrees centigrade. Shoot tips we can take out from the liquid nitrogen, and that shoot tips in ampule frozen in a liquid nitrogen, in the sense thawing process takes place here, ampule frozen condition takes place, again ampule stored in a liquid nitrogen. So you have to keep in mind, my friends, the things, the steps whatever involved in case of one. Here the word thawing is there, thawing in the sense the heating, again once again the things are there, and when we have to take out these shoot tips in the semi-solid medium for the further developments. So this is the little bit outline of a protocol for the cryopreservation of a shoot tip, and dimethyl sulfoxide is used here as a cryoprotectant and also use the liquid nitrogen. Now development of a sterile tissue culture, first step of cryopreservation is this one. Any tissue from the plant cell can be used for the cryopreservation. For example, that are nothing but the which type of tissue we can use in a cryopreservation, that is nothing but we can use meristems, we can use embryos of the plant, endosperm, ovules, seeds, cultured plant cells, protoplast, callus, all material, these materials we can use. Suspension cell culture in the late lag phase or log phase, or most these are the materials which is suitable for the cryopreservation. Now addition of, we have to keep in mind, already I have told you the substance is used to preserve the germplasm with the cryopreservation methodology, that are known as a cryoprotectant, that are known as a cryoprotectant are the compound that can prevent the damage causes due to cells by freezing or thawing. हे chemicals काय करतात, they are preventing the damage causes to the meristems or shoots or embryos at the time of their conservation, as a germplasm. So that is avoided by using the chemicals, or sometimes that chemical side effect is there of that chemicals. The freezing point and the supercooling point of water are reduced by the presence of a cryoprotectants. Again, you have to keep in mind the freezing point is taken. We know that frozen state is a cryopreservation methodology. Most important stage is there, and when we are using such a type of things, this cryoprotectants helps that the ice crystal formation is retarded during the process of a cryopreservation with the help of a cryoprotectant. There are several cryoprotectants which include the DMSO, dimethyl sulfoxide, glycerol, ethylene amine, DMSO, glycerol, ethylene amine, propylene, sucrose, mannose, glucose. These are the cryoprotectant chemicals used during the cryopreservation method to conserve the germplasm. Among these, DMSO, sucrose and glycerol are most widely used. Which are most widely used because they are very much suitable, easily available, and having a less side effect at the time of conservation of a germplasm. Generally, a mixture of a cryoprotectant instead of single one can also be used, which is a more effective cryopreservation without damage to the cells or tissue. Means we can use the mixture of these cryoprotectants, or we can use the single, but mixture of cryoprotectants instead of single. So mixture is very much, we have to say that, fruitful for successful cryopreservation as compared to the single using as a cryoprotectant chemical. Now freezing, as we know that freezing is the main core point of this cryopreservation. There are 4 methods of the freezing. Slow freezing method is there, rapid freezing method is there, stepwise freezing method is there, dry freezing method is there. So with these type of methods in cryopreservation steps we can use. So that are given here, which type of temperature, how much time we have to give for that one. So slowly frozen at a slow cooling rate, that is of a 0.5 to 5 degree DE GREE 5 degree centigrade per minute from 0 to 100 degree, and then transfer to the liquid one. You have to keep in mind 0.5 to 5 degree centigrade per minute, isn't it, from 0 to 100 degree, in the sense 100 ते 0 ते 100 डिग्री पर्यंत टेम्परेचर जे आहे त्यातलं जर वाढवायचं असेल, मीन्स मायनस करण्यासाठी, तर 0.5 ते 5 प्रत्येक मिनिटाला कमी करत जायचं आहे. अचानक टेम्परेचर कमी करायचं नाही. दॅट कॅन हॅपन, दॅट कॅन डॅमेज टू द लिव्हिंग सेल्स अँड देन ट्रान्सफर टू द लिक्विड. एकदा ते फ्रीज झालं की दॅट यू हॅव टू ट्रान्सफर टू द लिक्विड नायट्रोजन. Advantage of a slow freezing method is that some amount of water flows from the cell to the outside. This promotes extracellular ice formation rather than the intercellular freezing. आपल्याला माहिती आहे, हे एक्झाम्पल लक्षात ठेवा. जेव्हा पाण्याची बाटली फुल्ल भरून काचेची जर आपण फ्रीजरमध्ये ठेवली, तर त्या व्हॉल्यूमच्या पाण्याचं बर्फ होताना त्याचा व्हॉल्यूम वाढतो आणि बाटली काचेची फुटून जाते. कदाचित जर आपण फास्ट फ्रीजिंग करत गेलो आणि सगळं पाणी सेलमधलं त्या सेलमध्येच गोठलं गेलं जर्मप्लाझमच्या, तर काय होणार, ती सेल फुटण्याची शक्यता आहे. पण स्लो फ्रीजिंगमुळं काय होतं, काही पाणी ह्या त्या सेलमधनं बाहेर येतं आणि बाहेर आल्यामुळं सेलच्या आतमध्ये कमी पाणी राहिलं. कमी पाणी असलं की जजे त्याचं बर्फ झालं तरी तेवढं अकॉमोडेट करण्याची कॅपॅसिटी त्या सेलमध्ये राहू शकते. सो दिस पॉईंट यू हॅव टू कीप इन माइंड, माय फ्रेंड्स, व्हेरी वेल, इजंट इट? So this is of a slow stepwise freezing method, dry freezing. These are the methodology. Now storage, my friends, you have to keep in mind where to store all after freezing. In generally, frozen cells tissues are kept for the storage and temperature in the range of a -70 degrees centigrade to -196 degrees centigrade. -70 degrees centigrade to -196 degrees centigrade. That is most important. The ultimate objective of storage is to stop all the cellular metabolic activities and maintain their viability for a long-term storage temperature at -196 degrees centigrade in liquid nitrogen is a ideal one. काय घडतं, what things are going to happen? आपण ते स्लो फ्रीजिंग करत नेऊन पुन्हा त्यांना फायनल स्टोरेज करतोय ते फ्रोझन सेल्स. त्याला तर सेल्युलर मेटाबॉलिक ॲक्टिव्हिटी ह्या पूर्णपणे त्यांच्या स्टॉप केल्या जातात, पण त्यांची व्हायबिलिटी, कपॅसिटी टू जर्मिनेट, डेव्हलप इन टू ए न्यू प्लांट that we are preserving as it is at minus 196 degree centigrade storage. That you have to keep in mind. नाउ थॉइंग, थॉइंगला दुसरा शब्द मी सांगेन, दॅट इज ऑफ ए हिटिंग माय फ्रेंड्स. थॉइंग इज नथिंग बट हिटिंग. लक्षात ठेवा, HEATING, हिटिंग इज देअर. थॉइंग इज युजली कॅरिड आउट बाय प्लंजिंग द फ्रोझन सॅम्पल्स इन द ॲम्प्युअल्स इनटू ए वॉर्म वॉटर. इन द सेन्स आपण ज्याला फ्रोझन ह्याच्यातनं जर्म प्लाझम मटेरियल ॲम्प्युअलमध्ये घेतो, टेस्ट ट्यूबमध्ये घेतो, बॅटरी म्हणजे एखाद्या कंटेनरमध्ये घेतो, तिथं घेतलं की ते ॲम्प्युअल्स वॉर्म करायचे. किती डिग्री सेंटीग्रेडला? 37 ते 45 डिग्री सेंटीग्रेडला. बाथ विथ ए विगरस स्विरलिंग बाथिंग करायचं, त्याला आंघोळ घालायची त्या पाण्यानं दुसऱ्या वॉर्म वॉटरनं. लक्षात ठेवा. बाय दिस अप्रोच रॅपिड थॉइंग ॲट द रेट ऑफ 500 to 700 per minute occurs, and this protects the cells from the damaging effect. And ice crystal formation त्यामुळे त्याच्यामध्ये घडणार नाही, डॅमेज घडत नाही. ॲज द थॉइंग ऑकर्स, आईस कम्प्लिटली मेल्ट्स. ॲज द thawing occurs as the ice completely melts, the ampoules are quickly transferred to the water bath in the temperature 20 to 25 degrees centigrade. This transfer is necessary since the cells gets damaged if left for a long time in a warm 32 to 45 degrees centigrade. Isn't it? Yeah, that things you have to keep in mind. What we are doing step by step, you have to learn about the cryopreservation. आता लक्षात ठेवा काय केलं आपण. पहिल्यांदा आपण काय केलं, व्हॉट वी हॅव डन, द मेरिस्टेम शूट्स वगैरे ते घेतले. दॅट वी हॅव स्लोली वी हॅव स्टार्टेड टू make them a cool slowly, and finally after cooling we have put or store that into a minus 196 degree temperature. लक्षात येते. लिक्विड नायट्रोजन आफ्टर क्वाईट लॉंग गॅप or after quite long we have to say that storage, after many years we have taken out the shoot that taken into ampoules, and immediately we have heated up to 27 to 45 degree centigrade temperature वॉटर बाथमध्ये टाकलं, त्याच्यातून पुन्हा काढून घेतलं आणि रिकल्चर पुन्हा आपण त्याच्यापासून नवीन प्लांट तयार करतोय. दॅट यू हॅव टू कीप इन माइंड. मीन्स रिकल्चर इज दॅर. नाउ इन केस ऑफ दिस रिकल्चर व्हॉट वी आर गोइंग टू डू, थॉड जर्मप्लाझम म्हणजे ज्याला आपण हीट केलं, हे केलं, हिटिंगच्या पाण्यातून काढलेलं, दॅट वी हॅव जर्मप्लाझम वॉशड सेव्हरल टाइम्स टू रिमूव्ह द क्रायो प्रोटेक्टंट, द केमिकल्स वॉट एव्हर वी हॅव यूज्ड टू मेंटेन द वायबिलिटी, टू कीप द वायबिलिटी ऑफ ए germplasm as it is at minus 196 degrees centigrade, or at, we have to say that, zero molecular activity. This material is then recultured. पुन्हा त्याच्यापासून नवीन फ्रेश मिडियम स्टँडर्ड सोल्युशन आपण घेतो आणि त्याच्यामध्ये आपण ते वाढवणार आहे जर्मप्लाझम. दॅट इज अ रिकल्चर. नाउ हाऊ टू मेजर the survival or viability? The technique used to determine, my friends, to measure the survival or viability of a germplasm is here. Cells are staining technique. Cells are staining technique, TTC आपण वापरू शकतो. That is a trizolium tetrachloride, one of the chemical is there. Embryo आपण काही घेऊ शकतो, and that we can make it. पिंक एम्ब्रिओ झाला तर डेफिनेटली वी हॅव टू से दॅट व्हाईट एम्ब्रिओ असो, तो जर TTC सोल्युशनमध्ये टाकला 0.5% TTC सोल्युशनमध्ये टाकला तर कालांतरानं पिंक झाला तर आपण म्हणू शकतो दॅट इज व्हायबल. इव्हान्स ब्ल्यू अँड फ्लोरोसन्स डायसिटेट आपण वापरू शकतो. सो अशा नंबर ऑफ सेल्स ज्या ओरिजिन याच्यामध्ये ग्रीन झाल्या डिवायडेड बाय नंबर ऑफ सेल्स पर ऑर्गन थॉइंग मल्टिप्लाइड बाय 100, आणि जी आपल्याला रीडिंग मिळेल दॅट इज द मेजरमेंट ऑफ ए सर्वायवल ऑफ ए जर्मप्लाझम आफ्टर टेकिंग आउट ऑफ द फ्रोझन स्टेट, आउट ऑफ द क्रायोप्रिझर्वेशन. त्याची वायबिलिटी आपल्याला मेजर करता येते. सो थँक यू व्हेरी मच स्टुडंट्स. टुडे वी हॅव ट्रायड टू कम्प्लीट द व्हेरी इम्पॉर्टंट मेथडॉलॉजी दॅट इज द क्रायो प्रिझर्वेशन. पुन्हा एकदा मित्रांनो मी सांगेन, समजा एक प्रश्न विचारतो मी तुम्हाला बिफोर कम्प्लिटिंग दिस लेक्चर. समजा आपण एखादं छोटंसं जर्मप्लाझम रोपटं एखाद्या वनस्पतीचं घेतलं आहे. ते घेतलं कधी? आपण 2021 साली घेतलं आणि minus 196 डिग्री टेम्परेचरला किंवा क्रायोप्रिझर्वेशन मेथडनं आपण जर्मप्लाझम प्रिझर्व करून ठेवलं. कधी ठेवलं आहे? 2021ला. आणि समजा आपण 2021 30 लाख तेच रोपटं बाहेर काढलं तर त्या रोपट्याचं वय किती असेल? मला माहिती आहे तुम्ही सांगाल बरोबर 2021 30 9चा फरक आहे 9 हे उत्तर सांगाल. माय फ्रेंड्स दॅट इज नॉट ॲक्च्युली द answer of that your question, बिकॉज क्वेश्चन इज दॅट 2021ला आपण ठेवलं त्यावेळेला त्याचं वय जेवढं असेल 15 दिवसाचं तेवढंच आपण 9 वर्षानं बाहेर काढल्यानंतर सुद्धा राहील. विथ दिस थँक्यू व्हेरी मच. इफ यू हॅव एनी डिफिकल्टी आस्क.